Hello everyone, I'm Charlotte. So today we will have a look at what's called intraspecific variation in DEB parameters, which means a variation in parameters within the same species, so between individuals or between population of the same species. First, we will investigate why that could matter to our understanding of the species biology. And then we will discover a few methods that could be used to estimate, measure that variation. I need hardly tell you that speaking of a dog or a human being is hardly describing them all. Individual variation is widespread in living organisms, even clonal ones. So I also showed you here a few examples from salmon, lizard, or snails that show morphological or color variation, but that visual variation is actually also linked to differences in reproductive strategies or differences in habitat preferences. But the real question is, why do we care? Does it make any difference that a species is not an homogeneous group of individuals? So first, of course, we have what we usually think about as variation between individuals, which is variation in some kind of continuous quantitative trait. Here, for example, in a copepod, we can see variation between individuals in developmental and in brood size, for example, which are represented here as the histograms, the experimental data. And we can relate that as variation in physiological parameters, and for example, as DEB parameters, as we will see later. So for example, here, we can see how to reproduce that variation in the data based on variation in assimilation rate. And this variation in terms of population processes will add up, because here, for example, an organism with a higher assimilation rate will both have a shorter development time and a higher brood size. This way we'll have a higher lifetime reproductive success. But some traits, conversely, are not continuous. They are either binary with two options, or they can have a few different values. And sometimes this corresponds to very different, distinct phenotypes in the organism. To take an example I know well, here Atlantic salmon, we see that depending on the level of energy available early in life, in their first spring basically, they will have very different life history strategies. So some of them might be able to mature in freshwater in their first or second autumn if they have enough energy. And in that case, they won't leave freshwater, so they won't have to incur migration, higher mortality, but they will be much smaller also. Whereas individuals that do not have enough energy will migrate to sea, which will take them more time. They will risk higher mortality, but they will come back also larger, and so they will be able to secure spawning with a female, so on a reed. Which means that continuous variation in a trait, or here, for example, in energy reserve or acquisition, which can actually lead to striking differences in the behavior or in the life history of the organism, which might have important effects on the demographic processes. Going to that, here we have the example of a small community with a consumer fish, a planktivorous fish on the left, and then a top predator, a piscivorous fish on the right. And using DEB-based population and community modeling, the authors here studied the demographic processes between these two groups. So higher up, you can see on the first line what happens when there is no variation in behavior and physiology between the consumers, between the individuals in the consumer population. So what happens is that before the top predator is introduced, you have huge oscillations in the population. And then, so around the middle of the graph, you see the introduction of the top predator, which stabilizes the population oscillations and to a low level in the consumer population, and then the piscivorous fish achieves quite high population density. Then going down on the graph, you can see what happens with increasing variation in the consumer population. So what happens is that the overall oscillation in the consumer population are lower, but they are not that impacted by the introduction of the predator anymore. And that's for a good reason. It's that the top predator itself has difficulties invading the population. So as you can see on the right, the higher the variation within the consumer species, the lower the density of the predator here. So having a much more diverse populations of prey, of the planktivorous fish here, makes it much more difficult for the top predator in this situation to actually exploit this population and make a good living out of it in a way. Which means that even variation, and here that's mostly continuous variation at the individual level, will actually lead to huge differences when you go higher up in population and communities. Another problem with intraspecific variation is that one point of DEB-based modelings and other approaches, ecophysiological approaches, is actually to predict what will happen at large scales, both spatially and in time. And it can be difficult to extrapolate when there are strong differences between populations of the same species. So here, for example, in edible mussels, you can see that on the left, the prediction of models are quite good when they are applied to the population from which the parameters were estimated. So that's the two populations on the left in gray. So the absolute error, the difference between predictions and the observed values is quite low there. But when you go to other populations, then the predictions are much poorer. So for example, for other Mediterranean populations, so the gray one on the center, we see that the error is already increasing a lot. And then when we go to much further populations, so those are in South Africa, we can see that the error is huge then. So you have very much difficulty predicting what should happen based on parameters which were estimated in another population. Which means that you have to pay attention to that variation when trying to predict things on a global scale. And there is something a bit trickier and probably even worse in our mind. Is that usually when we try to estimate parameters on a species in the DEB framework, we don't have access to lots of individual data. We usually only have average data, so an average growth curve or an average time at first reproduction and so on. And this could actually bias our estimates because it happens that the relationship is not linear and that estimating a species parameter, a unique species parameter, based on average data won't give the same results as doing estimations on different individuals from the same species and then averaging the parameters we get. Okay? Which is probably, so the second method is probably the best one because it will actually reflect what the actual mean of the parameter in the population is. So that might be a bit tricky to gather, to understand. So I will give you an example here. We have these springtails and they studied the effect of cadmium on these soil organisms. On the left, they use what we usually do when we do estimation in the DEB context. So they did a group fit. which means that they average the size for each treatment group and time point. And so each curve you see is actually a growth curve for one treatment group with all individuals averaged. And you can see the difference in growth with decreasing growth with increasing cadmium. And from this data, they could estimate their parameters and also ecotoxicological parameters, such as, for example, here, elimination rate or the tolerance, so the higher level that the organism can live with. And then they tried another method, which is the one I told about before. So for each group, they did the estimation at the individual level. So you can see that each of the 6 graphs here represent one treatment group. And within each treatment group, you have a curve for each individual. And so they did an estimation for each individual. And then they estimated the average of all these individual parameters. And using these methods, the ecotoxicological value they get are much different. So you can see that the elimination rate is about 10 times or more than 10 times lower. And the tolerance is also a bit lower, which means that actually the effect of cadmium would have been underestimated by using the first method, so the group fit method, rather than doing an individual fit. So we will try to see how we can account for this. And to do that, we will first have a look at how physiology, and in particular the DEB model, integrates the effects of genes and the environment. I won't teach you much by saying that an organism's phenotype is a result of its genes and its environment. These can, however, play out in different ways. First, some traits that are extremely important for fitness are what's called canalized, which means that they will have the same value whatever the genes or the environment of a particular individual. Then some traits are entirely or mostly genetically determined. That's what you see at the top center, top middle. And there you can see that the environment has little effect on them. And one example is sex determination in mammals, birds, or some fish, for example. Then you have other traits that are mostly environmentally determined. So that's what you have on the top right. In that case, you may see that genes make little difference. So the blue and red genotypes have the same value. But the lines are not flat, so they respond to the environment and they have different values for different environments. And one example is sex determination in other groups such as chelonians and other reptiles, where temperature plays a huge role in sex determination. Then, for most traits, both genes and environment interplay. They could either act additively, which means that different genotypes will have the same response to the environment, although they will have different levels of the traits. That's what you have at the bottom left. So you can see that the value in a given environment are not the same for the red and the blue genotypes. The red one has a higher value, but their response to environment are the same. The two lines are parallel. Okay, so that's what we call an additive effect. Then there is what's called gene by environment interaction. And that's what we have on the right, the bottom right. In that case, different genotypes here in blue and red won't have the same response to the environment. So it could be a reverse response, as in the center, where the phenotype of the blue genotypes will increase in environment 2 compared to 1, whereas it will be the reverse for the other genotype. However, in some cases, it's not a reverse phenotype. It's just that one genotype will respond strongly to the environment, whereas another won't respond as strong. Okay, and that's what you have at the bottom right. Okay, that's the type of concepts we use in evolutionary ecology. But to go from this mathematical representation to the actual biology, we need something that will actually produce a phenotype based on these different influences, and that's physiology. And here I represented that through a DEB model. So the DEB model is one theoretical representation of physiology, basically. And here, well, we see, and you might know by now, that the DEB model takes some environmental influences, such as food and temperature, and use these to produce a particular phenotype in particular environmental conditions. And these phenotypes might differ between species. And that's what I represent as a genetic variation in the bottom. So that's variation in their parameters. Whereas temperature will only influence the rates, and we do that in a parallel way. And so with a given temperature, a given food, and a given set of parameters, so a given environment and given genotype, we will have a particular phenotype. This means that actually plasticity is built within DEB model. So remember that plasticity is a response to environment, so having a different phenotype depending on the environment. And that's actually what a basic DEB model does. Here, for example, we represented the effect of variation in temperature on the left, or food on the right, on magna growth. So you can see the average growth in the dark blue line, but then there is much variation around this. At a given age, for example, we will have a different length, which is a phenotype, for a different temperature or food level. And here temperature and food only varies between individuals. It's constant for a given individual. This can also be represented in another way, which is called a reaction norm. And reaction norm are the blue and red lines I showed you just before. So that's the mathematical representation of the response of the phenotype to the environment. And here I gave you the reaction norm to temperature and to food. And what you can see is that contrary to the classical representation I showed you before, which is a line, so a linear reaction norm to the environment, actually the response to temperature and food is not linear. And it also changes with age. For example, here we see that the response of temperature length to temperature, is stronger at high temperature in young individuals, but when the individuals are older, around day 40 for Daphnia, here, some individuals will already have reached their maximum size, and as temperature is influencing growth but not maximum size, then we see that the higher temperature do not have any more effect on length. So that's a red line above. Whereas the lowest temperature for those individuals which had a slower growth and have not yet reached their maximum size still has an influence. And so that changed the shape of the reaction norm. And actually, we can see that a bit on the right, although in that case, the shape, the function is the same. It's just that the scale changes. So we have seen now that DEB models integrate environmental variation between individuals from the same species. However, for now, it rarely explored genetic variation within the same species. So genetic variation in their parameters is assumed between species, but we don't have much data yet on differences within species. So here I show you one example, one of the rare examples of a study by French colleagues who had a look at the share of genetic variation in different parameters, and the one they selected because it was the most likely for this species, so that's European sea bass, was EG, so that was the cost of growth, if you remember well. And they found that the heritability for this parameter was 0.4. Translated, this means that 40% of the variation between individuals is due to the different genotypes of those individuals. We will come back later to what could be the remaining 60%. We now have data-based evidence that there is some genetic variation in DEB parameters within populations. Now, I want you to try a short exercise. Try to recall from the previous lectures you had what could be the effect of varying different DEB parameters on the resulting traits, the phenotypes. So it's about drawing the shape, the expected shape of the reaction norm for these traits, so the DEB parameters on the x-axis, and in relation to phenotypic traits on the y-axis. So that's what we just did for length on the y-axis and food or temperature on the x-axis just before. Here, I put you some DEB parameters. You see them at the bottom. And on the left, you have some traits. And I want you to try to relate these two aspects. Okay? So stop the video until you finish that exercise, and then we will try to see what could be the answer. Well, have you found some answers? Well, it's important that you try, because it will give you some intuitive feeling about how your model will respond to a change in parameters. So if you haven't, take some more time, else we will go to have a look at some answers found by different researchers. So first, here are some results we got in my team on length, for example. So I haven't shown you everything, and I will explain you why. So I only showed the response to PAM, so the assimilation rate, to kappa and nu. So kappa is the allocation coefficient and nu being the reserve conductance. So as you see, PAM has a positive effect on size, which is not very surprising. The more able you are to assimilate energy, the quicker and larger you will grow. Then for kappa, it's a bit of the same thing, and actually in that case, it's one of the rare situations where the relationship is really linear. So we see that with higher kappa, we have higher size because we allocate more to the soma. That makes sense. For nu, it's a bit different. We would expect that a higher nu will lead to quicker growth, for example. And that's not always the case. Why is it so? It's because at low nu value, increasing nu does make a difference, because it's actually this parameter which is slowing down the mobilization of reserve for growth. But once nu is large enough, it's not slowing down this process anymore. Okay? So growth is mostly slowed down by the difference between the energy available in the reserve and the cost for maintenance. Okay? Which means that the limiting parameter is nu only for very low value of the parameters, but once it's large enough, it's not limiting anymore. Which explains the very hunched shaped of the reaction norm. Okay? I didn't put the different costs like PM or Kg because they globally have a negative effect on growth. And also I didn't put the energy, well, maturation energy at birth or at puberty, so this threshold, because these do not directly impact growth. They won't have any impact. So actually, in that case, the reaction norm would be flat, no response. Now, let's turn to reproductive output. How does that work? Some effects are still straightforward. So PAM still has a positive effect, because the more energy you are able to assimilate, the more energy you can invest in reproduction. For some of the parameters, however, it's a bit more counterintuitive. For example, for kappa, here we can see that we have some kind of inverse U-shaped relationship. We would have expected kappa to have a negative effect on reproduction, because if you increase investment in the soma, so you increase kappa, we would have expected that to have a negative effect on reproduction. And that's what we see at high value of kappa. But actually at low value of kappa, we have the reversed relationship. And why is it so? Try to think a bit about it. You can stop the video if you need. Actually, what happens is that to have high reproductive output, you need to allocate a lot to reproduction, but you also need to be large to acquire a lot of energy. So if you don't allocate much to growth, so of course a larger share of the energy you have will be allocated to reproduction, but the total amount of energy you will be able to get will remain limited because of your limited size. Okay. So which is why we have some kind of quadratic relationship here with an increase and then a decrease in reproduction. Well, for other parameters, the cost, for example, we have a negative impact, which is still quite straightforward. And we also see that some parameters do not have much impact on reproductive output here. What about development time? So here, of course, some values will be straightforward again. So a quicker development with higher PAM, which translates into a longer development time at lower PAM. So that's what the relationship is reversed, it's negative. So it takes you less time to reach the same stage with a higher PAM. And we have the reversed relationship with the different costs, like PM or kg, for example. Then here again, we have that kind of hunch-shaped relationship for nu, as we found for both length, reproductive output, and here development time, although it's reversed. And we also have that quadratic relationship to kappa for kind of the same reason, okay? Because you have to allocate a large portion of energy to reproduction to be able to mature. But then if you're not big enough to acquire a lot of energy overall, it will have a negative effect on your development time. So we've seen how these different parameters could actually translate the influence of genes on different phenotypic traits. We've also said that genes and environment could interact, which means that the relationships are not parallel, and thus the effect of the genotype won't be the same depending on the environment, or conversely, different genotypes won't respond in the same way to environmental variation, said in another way. So that's inherent in the model. To give you an example here, we know that the Arrhenius temperature actually translates a reaction norm to temperature. And so it's the interaction between temperature, the ambient temperature, and the Arrhenius temperature, the parameter, that will explain or predict the phenotype. Here, for example, we have the variance in length, in body size, due to variation in the genes, in Arrhenius temperature, but at different ambient temperature. And we see that the responses of the different genotypes are not the same. For example, there is little variation between genotypes, so the variance is close to zero when we are close to the reference temperature. Because if you remember well, the Arrhenius relationship is equal, Arrhenius corrector is null, so it doesn't change anything compared to the reference parameters when we are close to the reference temperature. by definition. Whereas when we're further from the reference temperature, so for example here at 0 or 6 degrees, then in that case we have huge variation. So the variance can be very high and it decreases with age at this temperature, which means that there are a lot of differences between the different genotypes. So that's a G by E interaction which is actually included already in the model, in the DEB model. However, we could also think about how plasticity could occur at a lower level traits. So it's not plasticity in terms of morphology or life history traits or behavior in response to environment, but it would be plasticity of the DEB parameters themselves, and that we have little information about. So we could think that, for example, organisms that would have been exposed to different temperature, different food, or different other factors from the environment at a young age would have different values of the DEB parameters later on. And that has been little explored. That could also go through epigenetic effects and parental effects, where actually the environment of the parents could non-genetically influence the DEB parameters of the offspring. Just to let you think a bit, we saw previously that heritability for EG was 0.4, which left us with 60% remaining variation between individuals in that parameter. So what could that be? It could be error in measurement error, or due to stochasticity in the system, because all individuals might not have exactly access to the same food at the same time, and they might not be exactly at the same temperature in the tank and so on. But then it could also be plasticity. So it could also be that those different individuals had different parameter value due to the environment they were subjected to or the environment their parents were subjected to. Now that we have explored the theory of individual and population variation within species, we will go to methods that could be used to estimate that variation. And first, we will have a look at variation between what I call subspecies. Here, by subspecies, I mean any group within a species. So that's a very broad definition, not only the taxonomical one. It could be taxonomical definition, so actual subspecies, but it could also be different population within a species, or it could also be different sexes within a species, for example. So first, one option, which is straightforward, is to estimate parameters for each subspecies, its group, as if it were a separate species. Okay. So in that case, we don't have to make any assumption on the variation between species, so which parameters are more or less variable between subspecies. The problem is that this method is quite sensitive to differences in the completeness of the dataset for different subspecies, so that differences in parameters between groups could actually stem to differences in the quality of the data. It's also difficult to identify and quantify which are the biologically significant differences, and there is no way to build a credibility interval for delta in that case, at least with numerical optimization methods. So another option, which is also used between species, between closely related species, is to do a joint estimation. So we actually estimate some parameters as equal between the species and others are more or less different. So the first option, the older existing one, is some kind of all or nothing choice, where we decide that some parameters will be equal between groups, or the others will be different between groups without any constraint. We can add some kind of covariation rule in that case, so that the parameter space will still be meaningful, so that, for example, the maturity levels will increase together, okay, in parallel. There is also well-documented covariation among species between these maturity levels and the zoom factor. So that can be done within, for example, the DEB tool set of functions, this kind of joint estimation. More recently, there was also another option that was proposed, kind of intermediate option, which is a use of a loss function to which a new part was added, so a kind of augmented loss function. So I just want you to stop the video here and try to recall and write down on paper what is a loss function. So you have heard about it in the previous lectures normally, and so I wanted to try to recall that. Do you remember? So the loss function is a bit similar to the likelihood in statistics. It's some kind of integrative and weighted measure of the difference between the observed data and the predicted ones. And so minimizing the loss function ensures that you will get the best possible combination of factor of parameters to explain or to reproduce your data. What does the augmented loss function do? It adds a parameter that will not only penalize the difference between predicted and observed, but also penalize the variation between groups. So if you have a weight in the loss function which is 0, so 0 means you don't use the additive, the augmented term, then variation between groups is not penalized for this particular parameter. For example, in the graph here, it's nu, which means that you come back to the previous situation where this parameter could freely vary between groups. But then you could increase the weight, and by that you will increasingly penalize the variation between groups. And you can see that by varying those weights, you actually at some point reach a threshold in the coefficient of variation you have between the values, and then also in the loss function, which means that you can use this weight and find the optimal weight to find what should be the distance and the constraint in the variation between groups for that particular parameter. So I won't go much into these methods because they were already well explored and defined between species, and they are quite similar. Okay? Just to finish, these joint estimation methods, with or without an augmented loss function, they open up the possibility to estimate the difference between groups as a parameter itself, and thus to produce a credibility interval for that difference. And even more interesting, they use information from the data-rich group to improve the estimation in the data poor groups. Okay? So they partly solve our problem of the differences in data completeness. Also, their main problem is that they require more assumptions on which parameters are constant or variable between groups, although using the augmented loss function partly solves this because it allows a more continuous variation between groups. Now, we will come to methods which are a bit more recent, most of them to try estimating individual variation in their parameters. So the first method I will present here is using the distribution of one trait, one phenotypic trait, to deduce the distribution of one DEB parameters, which is one I use this OAT formulation here, which means one at a time. What the authors did in that case is that they tried for different DEB parameters to find the one that would best explain variation in brood size or variation in developmental times. So you can go back to the slide on the copepods at the beginning, because that's the variation in traits they used. Then they used the reaction norm we've seen in part 2 to estimate, by fixing all parameters but one, which value of the parameter would give each individual value of development time or each individual value of brood size. And they obtain in this way the histograms you have in blue here, which are the distribution of PAM or the distribution of XAH, which is actually a coefficient which is applied to all maturation thresholds. And this way they could both had an idea of the value, the average value for the species, but also, as you see, how this parameter could distribute to explain the observed variation in the phenotype. And the other larger graph is an exploration of different distribution, different shape of the distribution. And they actually concluded that although the normal distribution could be used especially for PAM, the log normal better explained the observed variation in trait. One advantage of this method is that it's possible without having longitudinal data for each individual. Only the distribution of one trait would be sufficient. It also helps identify the most relevant DEB parameters, so the one that may vary the most in that particular species. However, it assumes that only one parameter explains all the variation, or at least most of the variation, and it also relies on only one source of information, one trait at a time, which might be problematic if there is lots of measurement error on that trait. Another way of identifying which parameters are more likely to explain the variation within one species is to use the relationship between traits. So here, for example, in European sea bass again, they use the relationship between reproductive output and size. They examined the relationship between these two traits that was generated by just age variation between individuals or environmental variation in food and temperature, and then DEB parameter variation between individuals. And they found that the observed relationship between reproductive output and size was best explained by either temperature variation between individuals or variation in PAM, in assimilation rate between individuals, but not well by the other parameters. So this approach is interesting because it's possible, again, without longitudinal data. You just need to know the broad relationship between the traits. Actually, you don't even need precise individual data. And it helps identify the most relevant parameters. So it's another way, and it could actually confirm information obtained by other methods. However, again, it assumes that only one parameter explains all variation. It's again a one-at-a-time approach. And it does not really estimate the parameter distribution. So we just know which parameter is most likely to vary, but we don't know what its values are within one population. Then other methods will actually use individual fit. So they will actually fit a DEB model to individual data, which is what we should all do usually, but we can be limited by the data available. So I come back here to the springtail and cadmium study, and this is only the data without cadmium. So just to show you how they did to estimate the parameters. So in that case, they fitted a von Bertalanffy growth curve on each individual. And doing that, they obtained the parameter space, or the set of parameters you can see on the right, between the growth rate and the ultimate size. Which is okay in that case, because you only have two parameters. But it can get much more trickier when you have more parameters and thus much more freedom in your parameter space. And then in that case, you can of course do statistics like producing a graph here or estimating the univariate distribution and so on of your parameters. In the next example, they actually fitted a full DEB model. But instead of using a covariation method where they estimated all parameters together, as they did for the von Bertalanffy growth. Here, they simplified the problem by using again a one-at-a-time approach. So they fixed all parameters to the best average species parameter value. And then, based on that, they varied only one parameter and tried to fit the values for each individual, so different growth in length and growth in mass value for each individual. And doing that, they obtained different errors between the observed values and the predicted ones, depending on which parameter they chose to vary. So on the right here, you can see that they found huge errors if all the parameters were constant between individuals, which indicate that there was some heterogeneity between individuals. And some parameters were almost equally bad. So nu, for example, kappa, or varying simultaneously PAM and the threshold for birth and puberty did not improve the error much. However, some parameters were able to reduce the error in the model. And that was the case for PM, for PAM, and for EG. And in that case, they used the distributions they obtained for these parameters to estimate their heritability. So they decided that EG was the most likely viable parameter. And that's how they estimated the 40% heritability we've seen before. In that way, so using many traits and estimating a model on one individual at a time, you can estimate variation in several parameters at a time, so as they did with the springtail study. However, you require longitudinal data. Usually you would require individual data for several traits, and in particular for size traits, for example. And these methods are very sensitive to error in individual data, of course. If you don't have that many individuals, that's even worse. And they are quite sensitive to identifiability issues, which is why the two examples I showed you either concern only two parameters. So they used von Bertalanffy growth, which is actually a simplification which can be reduced to a DEB model but at constant food and temperature. Or they use a one-at-a-time approach. So they decided beforehand which parameters were viable or not, and they tried each of them. So they did not estimate them simultaneously. Another approach which could be used is a more statistical one, so using likelihood approaches rather than loss function approaches. So instead of doing numerical optimization, you would actually do some statistical estimation. The advantages of such approaches, which have been used for estimating species-wide DEB parameters, but not yet individual ones. The plus are that each parameter variance and the covariance between parameters is directly estimated as a new parameter in the model. So you don't have to do two-step. First estimate for each individual and then calculate the variance between individuals, which is more sensitive to error in the individual data. These likelihood approaches, in that case, they better account for that kind of error. However, they require individual data on several traits, again, so that's the same problem as what we've seen before. They also require assumptions on parameter and error distributions. Not for kind of binary traits, so age at a particular transition does not need a distribution, because actually the underlying distribution is when it will reach a certain maturity level. But for size, for example, for the number of eggs, you need a distribution function. And then, because of all those variances and covariances, the number of estimated parameters may increase very rapidly. When you do individual fit using an optimization method, you might already have difficulties doing the estimation for all parameters correctly. And then in that case, actually with likelihood approaches, you're adding more parameters by having to estimate the variance of the DEB parameters or the covariance between them. And so that might be quickly computationally problematic and might lead also to identifiability issues in that case. Okay, so now we're done, and I would like us to sum up a bit what we've seen today. Okay, so what should we keep in mind? First, we discovered that there can be risks at ignoring intraspecific variation. First, it can lead to poor predictions in some populations or communities, and it can also bias our species level estimates and prediction, so that does matter. Then we had a look at how DEB-based models combine environmental and genetic sources of variation. We recalled the kind of reaction norm that we can generate and linked these concepts to classical concepts in evolutionary biology. Finally, we listed some methods for estimating intraspecific variation, and I would like to outline 2 points. First, assumptions on variable parameters may help improve identifiability, but they do increase the risk of errors. So you can do that in an all or nothing way by saying these parameters are variable and those are not, or you can find more refined way to constrain this variation. But those decisions you make, if they're wrong, of course they will have a large influence on your results. Then, measurement errors and intraspecific variations still remain difficult to tell apart using the current approaches. So finally, I would like to thank you all for your attention, and I would also like to thank the different researchers and students who contributed data and allowed me to show them today. I'm looking forward to meeting you almost in person and discuss this with you soon.